You are allowed to have your lab notebook. That's another reason why having your lab notebook with approved hypotheses and objectives and stuff is really useful, because when you want to make sure you understand what was the point of a particular lab, if you have your lab book done and it's done well, you've got the answers waiting for you. So on this particular exam, you still aren't allowed to use like random crib notes and things like that, but you are allowed to have your lab notebook, which has your discussions, which has all the basic information about what we were doing, what we found, and all of that. If you've written your discussions well, it should clearly explain all the important parts of what the lab was about. So the actual questions on the lab exam are going to be a mix of, it could be theoretical things, in the eyeball, which part of the eyeball is doing the transduction versus doing the focusing or something. You should know, like, the theoretical underpinnings of things. But then there'll also be like, I could have a picture of like, I know this one like just kicked people's butts. The osmometer. Can have an osmometer lab. Here's an osmometer, and I'll say, I'll give the fluid color just so you can see it. I'll say, here is my osmometer, and this is distilled water here. And this is at equilibrium. This is in a stable situation here. It's not changing over time. And then I could ask, can my red solute penetrate the dialysis tubing membrane? Yes or no, and how do you know?
It can't, because if it did, it would be distilling out into the H2O, and instead the water is going into the pipette.
Yeah, so the fact that we have equilibrium, water up the tube fighting gravity, that means there's osmotic pressure happening, which means we have to have a gradient between the inside and the outside that's maintained. If the solute could leave, it would. It's like pure water out here. It's in here. It would just go out until it's equal in and out, and there's no difference anymore. There'd be no osmotic pressure, and the level would just be at the level of the water rather than up the tube, right? Does that make sense? So things like that. Like if you understand the idea of osmosis and how this osmometer works, that would make sense. If the osmometer was still confusing, or osmosis was still confusing, then this would not make as much sense or be as easy to figure out. So something like this could be on the exam, just a picture of an osmometer, some question that makes sure you understand the core concepts that are going on. Or it could be questions about what we found and what it meant and why that made sense. There could be graphs of like, here's a person's jump in their height, and what can you tell me about this, and does it make sense, and how does it relate to your, I'd have units and stuff on it. But there could be graphs. There could be graphs that are familiar from the kind of data that you've been working with for one lab or another. And you've got to interpret that based on what you know about the relationships we've been learning about, maybe even putting it in the context of what we found. because you'll have that in your lab book. You'll know what you found and you can compare. You know, if we go all the way back, we can just start back from, you know, there's the Chem Review Lab. You need to make sure you understand what were some of the take-home messages for this Chem Review Lab in the very beginning?
That electrolytes conduct electricity.
Yeah, that electrolytes, when you, especially when you put them in water. And why? Why is that?
Is it because the hydrogen ions break apart into the water?
Not hydrogen ions.
The ions.
Yeah, it's usually they're ionically bonded things like, you know, sodium chloride. And so you get like your anions and cations independent and floating around. What happened when you tried to dissolve did it work if you put it in oil?
Oh, that's a buffer.
No.
It didn't dissolve.
Oh, it didn't dissociate.
It didn't dissolve or dissociate. Why was that?
You don't have the same hydrogen bonds that the water has, pulling the cations and anions apart.
Yeah, exactly. It was a non-polar solvent. So there's no charge imbalance in the, you know, solvent molecules to tempt the ions to have new little liaisons and go find their own adventures, right? Right. If you're just in a non-polar solvent, there's no charge imbalance around you. So there's no better game in town than just staying stuck onto your sodium chloride crystal. Whereas if you're in water, there's all these polar molecules floating everywhere. It's like, well, this is fun, but that looks fun. And you go off and, you know, start floating around as an independent ion, which can then carry charge as electrical current. So hopefully and again, these concepts are going to continue being at the core of things till the end. So these are useful things. Hopefully by the time you walk away from this class, this is really going to feel like, oh, okay, I got it. What about, what other things did we look at in the class? There was whether things conduct electricity. What other thing did we look at that was important?
Buffers?
What's that?
Buffers?
So the buffers, that was, yeah, so let's stick to the part that we're talking about first with we will get to the buffers. That is the other thing we did look at, but just to try to finish.
I looked at pH.
No, that's more about the buffer part. So in addition to conducting electricity, what else could we describe the behavior of salt when you put it in water? What happens to it?
It completely dissociates.
So it did dissociate, and and that's something you can tell with the electrical behavior. What else did it do that you can tell just visually by visual inspection?
It dissolved.
It dissolved. The crystals disappeared and turned into just a clear solution. Right, and then we know that probably something more happened with dissociation, because when we try to do the electrical stuff, we saw the electrical behavior. But in terms of just looking at it disappear, we know it's dissolving. You know, again, did it dissolve in the oil?
No.
No.
No. The whole like dissolves like stuff. I mean, what else dissolved that we looked at?
Sugar. Glucose.
Yeah, the glucose. So, I mean, a big part of this lab was this idea of dissolving versus dissociating. Remember that? Like you can have sugar and salt superficially. It looks like they're doing the same thing. They're both crystals are disappearing and going into solution. They're dissolving. And that's true because you have a polar solute and a polar solvent. And then we also saw there were differences, based on this dissociation and the presence of ions with the electrolyte. So things like that, like you want to be able to go back and remember what we found, what the take-home messages were, what the evidence was for the different things. So, I mean, part of a lab practical is like practical, like how did we actually know whether or not something conducted electricity?
Put the electrode in
And
it buzzed if there was electricity present.
yeah, we use that.
Right.
If we could complete the circuit, if the liquid could complete the electrical circuit and you could hear the buzzer buzz, then you knew that the solution could conduct electricity. So there's things like that. Then there was also the pH part of the lab. What was the important part of the take-home messages for the pH part?
That as you add acid, it becomes more acidic, and as you add base, it becomes more basic. But if you have a buffer, there's little change.
Yeah, it was the whole point there was how labile pH was, especially when you're adding in strong acids and bases. And yeah, how dramatic, right? How dramatic the behavior changed when we use buffers. Because right, it's gonna be quite a bit later. It's gonna be another month and a half. We're gonna be doing acid base balance and stuff. But buffers are gonna be super important part of that story. So it's important that you understand what a buffer is and kind of the basic idea of why it works. So, so that kind of part, that stuff's important, kind of what we found and what the implications are. Let's see. You go back. There was the jumping lab. For a lab like this, pretty much for all the labs that have hypotheses and stuff, you should make sure you know the objective, right? What were the independent, dependent variables? What kind of relationship were we trying to figure out? And then what did we actually find? And kind of what was the data and the interpretation of the data? If I could ask you, I could ask you just questions about what was your hypothesis? Again, because you've got your lab book in front of you. Like, what was your personal hypothesis and was it supported by the data or not? That would be a fair kind of question to ask. Kind of looking at your process and your interpretation of the data. Jumping lab. Oh, let me get my.
Would you have told us if we were totally off in our interpretations? In our lab?
Of the ones I've graded so far, yeah.
Okay.
Yeah, no, so a number of people are probably very aware that I can be quite voluminous in my discussion of like, this doesn't make any sense to me at all. So.
Okay, good.
All right. Let's see. Osmometer Lab, we just kind of talked about. Fluid balance and RBC Lab. What were the main independent variables for this one?
Type of solution.
What do you mean?
And the
A little-
concentration.
So one was the concentration, and what was the other?
Whether it was penetrating or non-penetrating.
Yeah, the type of solute, and the thing that really made a difference was whether that was penetrating or non-penetrating. Right, and you should be able to go and look at what we found, what it means. And also, again, since it's a lab practical, I would feel perfectly fine putting some pictures of test tubes on the screen for you, and you have to interpret what happened. What does it mean if the supernatant is all red?
Lysis.
Green.
Yeah, right. So that would be hemolysis. That means that cells broke open and released the hemoglobin up. If crenation, what would crenation look like?
The cell was like shriveled up.
But
Was
you're
it
not,
smaller?
but you won't see that. What about in the test tube?
Smaller pellet size.
Right, or a pellet. I mean, if you don't have anything, let's say you didn't even have anything to compare it to. It's just a test tube with some hypertonic solution that's going to crenate the cells, and you put the cells in there and you spin it down. What are you going to find?
Clear supernatant in a small pellet, red
You're going to
pellet.
see a clear supernatant because nothing exploded. So all the cells just pulled down, and a pellet because the cells that are there all got pulled down to become a red pellet. Right? If I have the luxury of comparing exact same amount of blood that went through the same procedure, but one in a hypertonic and one in an isotonic solution, I might be able to notice a smaller pellet with a hypertonic solution. They're going to be smaller. Under a microscope, how could you tell that they're crenated?
They look spiky.
Yeah, then they're going to be all small and spiky and shriveled, as opposed to a classic looking blood cell that looks kind of more like a puckered frisbee, or hypotonic, where it's going to look kind of like overinflated if it hasn't popped even. So make sure you go back on that lab and kind of understand the basic concepts. And the difference, what's the difference between like isoosmotic versus isotonic? So say that louder. Hello? Oh,
Is like isosmotic, like how cells move through the cell, like if they can move back and forth between the cell or move out of the cell.
no, an isoosmotic solution means what?
There's no movement of water, or no net movement of water.
Same osmolarity.
Means the same osmolarity inside and out. Be 0.3 inside always, so it means anything that's 0.3 osmolar is isoosmotic. Right, so you really do need to get these definitions. Back to Sienna's thing, what would it mean if there's no net movement of water? What's the term we use for that?
Isotonic.
That'd be isotonic. There's like the same pressure on either side, the same osmotic pressure. And often isoosmotic is going to be isotonic. But is that always true?
Only if it's non-penetrating solute.
Exactly. Only if it's non-penetrating. When it's penetrating, we saw that the solute moves in and then water follows, and all of a sudden it's no longer isotonic. In fact, what do we call that when water moves in?
Hypertonic?
Or hypotonic?
Hypotonic.
Oh, hyper is moving out, right?
Yeah, hyper is moving out. I mean, again, I use, it's kind of a similar mnemonic. I always think about my lips and the sounds, like hypo, it's like, it's all like big, like it's the O is like getting all swollen because all the water's coming in. Like the hyper, it's deflating, the water's leaving. You know, I'd say come up with mnemonics. It's not, you're in good company if you come up with silly mnemonics to remember things, because that's how everybody does it. They come up with silly little ways to kind of try to remember things and keep track of things. If you just try to like remember it and hold on to it, you'll probably forget it. But if you have some silly like image in your head of some little cell going, it's much more likely it's going to stick in your head. So RBC Lab, that's an important one. Enzyme Activity Lab. This one,
I may have a mnemonic to help people with the hypo and hyper. The way I remember it is I think of hyper children, you want them out. So that's the way the water goes.
okay, so that works too. So yeah, like I said, coming up with mnemonics is a really good way to remember these things. I came up with mnemonics when I was still in college that I still use when I'm teaching things. I'm in front of the classroom. I'm like, what was it? Oh yeah. And I just, so yeah, they can last a lifetime. Enzyme Lab. This one confused a number of people. And as I've gone, some people have come into office hours and I realize, you know, and this is good. It's actually great when people are confused and come into office hours and we actually have a chance to straighten things out, because that's what the whole point is. It's like you're learning it and it's confusing by definition when you're learning things, you don't know it. And then you go through the process and learn it, which again, that's why I encourage people who are confused to, you know, to take that extra step to kind of get on top of it. You know, so some people have, I've been appreciating, some people have been doing that more. Enzyme Lab, what was the independent variable and dependent variable?
Independent was the substrate concentration.
Exactly, the substrate concentration.
Dependent was the reaction rate.
The reaction rate. You know, and ultimately, in order to find that relationship, we would end up with some graph that was reaction rate, which we measured in mils of oxygen per second, and substrate concentration, which was, you know, just kind of percent, 20%, 40%, up to 100%. You know, what kind of relationship did we find?
Reaction rate increased as substrate concentration increased until a saturation point was reached.
Yeah, so it's in general, it goes up more, higher concentration, faster the reaction. And I'm pretty, for most of, I think I did get it about right. So at the highest levels, it doesn't keep going up anymore. Even though you're increasing substrate concentration, it's not going any faster. Again, what would cause something like that? Behavior like that?
All the enzyme being tied up in the reaction.
Yeah, exactly. All the enzyme is busy. That's what we called saturation, if you remember. So the thing that gets people confused on this one is how did we actually get this graph? How did we actually get a measure for reaction rate at a certain substrate concentration?
Measured the slope of each trial at the point when the reaction was running smoothly or running quickly.
Exactly. We had to do a different kind of graph. We did time in seconds versus mils of oxygen produced. And it kind of goes, and we kind of measured the slope. That slope is the reaction rate that we put as a dot on this thing for some whatever the concentration of the enzyme is for that trial. Again, what people, I think people get confused because these look similar. Like if let me draw it bigger. I think this is worth drawing bigger. You know, it could be, this is trial 1. This is like, 20%. And this is 40%. And 60%. This be trial 3. And 80%. And this is going to be time in seconds versus mils of oxygen. On this graph, what does it mean when the graphs become flat?
Total saturation. Like of the enzymes, right? They're all being used up.
Right, so that you can't see saturation on a graph like this. Actually, you can, but not from looking at that. Right,
Isn't
The enzymes are used up.
The reaction is slowed down.
not just slowed down. If this is a straight line, the reaction is stopped, right? Time is continuing on. The amount of oxygen is not changing at all. The reaction's over. You've broken down all the substrate. There's no more hydrogen peroxide to break down at all. So you could wait until the cows come home and it's just going to be a straight line because there's no more substrate. There's no more reaction to happen. Where is my evidence of the reaction rates happening in this graph?
the slopes converging?
Well, the reaction rate is going to be the slope of whatever this, when it's really going, right? So like this is what I use to make that other graph. If I want the graph of substrate concentration versus reaction rate. I would measure the slope for this one here, and that would give me a dot for 20%. I would have to measure the slope for the 40% one there, and that would give me the reaction rate for 40. I'd have to measure the slope, at my 60% here, and that would give me my dot here. Right, and then when I got to 80%, what do I notice about the slope on the 80% compared to the 60%?
Barely changed.
It's pretty much the same. So I'm going to start seeing like, oh, but notice this graph down here flattening out, it's totally different than the graph over here flattening out. Here, my independent versus dependent variable is flattening out due to saturation. Here, I'm looking at a particular trial of oxygen over time, and it's flattening out because the reaction's over, right? So is there anybody like confused about that?
I was confused about what the graphs were that you put in the data for us.
The graphs, I think it's like this. It's just time versus mils for the different trials. I'm pretty sure. Is there something else in there that I forgot about?
I don't know, it's just the units didn't really make sense, so there wasn't labeling on the axes. So there were two different ones that looked pretty different, but I couldn't tell what they were.
Wait, so I have to take, there might be stuff in there I didn't realize was in there, because that doesn't sound like me to not label axes. And did you open it up in Excel rather than in the browser?
I'm pretty sure.
Okay, I'll take a look at that. That's surprising to me. I'll have to figure out what the heck happened there. So it's 12:30. I'm happy to keep going. I realize it's 12:35, but I think we should just, I'll keep talking, and then I'll also this will go up on the YouTube. I am going back to the module now, because now I'm worried that I put something up there that's confusing to you guys. Let's see. Hold on. I have, gosh darn it. Hold on one more second. Wait, where did I put? Oh, there it is. There's Enzyme Lab data. I'm opening it up. Let's take a look at it. So here's just the trial 1, trial 2, trial 3, trial 4. No, it just says time in seconds, oxygen evolved.
I must have opened it up in a different one because mine didn't. Yeah, it looks different.
Yeah, no, so you always have to open it up in the native application, or else, yeah, the little preview things in the other programs that'll show you kind of sort of what's in there, but it doesn't give you the real thing. So, all right, now that I'm relieved about that. Okay, let's continue. The Nerve Stimulation Lab. This is another one where you have to be careful about keeping track of what is the independent and dependent variables and how we measured them. What was the independent variable for this one?
Body location.
So one was body location.
And frequency of stimulus.
The other one was the frequency of the stimulus. How fast we were pulsing the little electrical current across the skin. And then the dependent variable.
Threshold.
Yeah, that sensory threshold. And how did we measure that? How did we figure that out?
By changing the frequency and gathering what number of amps were required.
So I would say that's confusing. There's something much more straightforward and clear way to say how we did it.
So like if we took like 1.0 milliamps as the threshold, that was at 0 hertz, right? That was like the single stimulus. And then we would minus the 0.6 from 1.0.
Right. Again, you're getting more complicated. I want just what is the technique in any particular condition. You're maybe at some place on the body, or the frequency is set, or the stimulus is set at a particular frequency. So for some condition of the very independent variables, you are going to measure the dependent variable. How do you do it?
You start really low and then slowly change it until you feel something.
And change what?
Well, we were changing the frequency.
The stimulus?
So, and what was the stimulus?
The
Current?
The amount of current.
The current. So it's the electrical current, the milliamps, right? I think I want to make sure people understand the milliamps is kind of like the magnitude. Frequency is how fast the pulses are coming. But I think people are getting confused. Like I can have, 2 hertz. Like it's like that at 2 every second. I could have 2 hertz at 1 milliamp, 2 hertz at 2 milliamps. I could have 2 hertz at 3 milliamps. So the magnitude in milliamps is very different than the frequency, like how often is the pulse happening? Right, and so our sensory threshold is what is the minimum current, the minimum number of milliamps in any particular condition that you can just barely feel. Does that make sense? Right, so you might set the thing to 2 hertz and you're measuring on the finger. Those things just stay. Now you start, obviously you have to start with some milliamps that's smaller than you can feel. Didn't feel it? Okay, jack it up. Okay, do you feel that one? Nope. Ooh, I feel it now. Okay, so there, let's say that's 3 milliamps. That's going to be my threshold. That's the minimum value of current in that condition that I can feel. So that is the sensory threshold for that particular condition at that frequency on that particular body part. Is that making more sense? Yes, no? So for those of you who are left, just tell me if I should move on, or if I want to, you are my kind of ears, because people who have dipped are not going to be able to say, I don't understand it. So I want to make sure I leave a recorded record that's actually going to be helpful for people.
I'm fine.
It's good? Okay. All right. The rest of the labs are the ones that we've just been doing. General Stimulation Lab. Right. Normally, this is like a fun one to just kind of go over all together, but like
Is
I
this a
said,
general senses lab?
yeah, oh my God. Yeah.
And I think did you skip the reflex lab also?
I kind of did, just because it one seems pretty straightforward, but do you have questions about that one?
I don't think so.
I mean, you should definitely know the different reflexes. Yeah. Let's see. But for the general senses, I'm going to go bring up the data. Where is it? Bah. I'm not there yet. Here it is. The 2-point discrimination is, at least when I first did this kind of stuff, it was very surprising to me. Not, fingertips and lips, that's not that surprising. Those had very fine discrimination. You could have two things really close and still feel them. Like your face. Your face, most people think of your face being so kind of sensitive. 1.5 centimeters, that's like over half an inch. You can have two pins over a half inch apart, and you have no idea that it's two things instead of one. Like this idea that you have such a limited capacity to actually detect things on your skin is a little non-intuitive. I think you just kind of assume, oh, I know my fingers, I bet you the rest of my body's kind of like that, but it's not at all. You get to the back of the calf, you have two things that are over an inch apart from each other. You have no idea that it's two things instead of one. That's part of the reason why I think it's worth doing this kind of experiment. It's just to get a reality check. Like if you just heard it in a class, you might not believe it until you actually did it to somebody and realize, ooh, that's weird. What is probably responsible for such a difference in 2-point discrimination at different parts of the body?
The sensory receptors.
Yeah, kind of the density of the little sensory receptors in your skin, right? If you have a very dense array, then you can detect things that are very close as being separate versus just the same. If I'm in my finger, I've got all these little receptors sending messages to my brain about touch, 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 touch. If I'm on the back of my calf, it's kind of like this. So if I have a pin here and a pin here, it's like on, off, 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 on. It's like ching, it's obviously two. Your brain knows without having to even struggle. You put two here, and all your brain knows is like, it feels something in this vicinity. It has no idea if this would be the same if it was one big thing or a bunch of little things. All it knows is that there's stretch on that particular touch receptor on that area of the skin. So the density is kind of the same thing that was going to be responsible for having the lower thresholds on the fingers versus on the back of the calf, right? And it's the same thing about the touch localization, right? If I took the black pen and I said, okay, here's the dot, and you're trying to figure out where is it, and you're reaching around, you can get pretty close because you have pretty good information about where the original pen was and where your current pen is and try to match them up. If you're on the back of the calf and do that, it's like, for all you know, it's somewhere around there. And then you put your other pen down trying to find the same spot, and you have a lot of slop. And hopefully people also realize, if the people who've done it, it's very weird. I know for me, it was bizarre how, again, you are touched and you're sure you're touching the exact same spot and you're a half inch away, because you're limited by the resolution of your touch receptors in your skin. So, whoa, my light just came back on. That's weird. I have no idea why it came back on, but I'm glad. So, did anybody here have a chance to do the thing, putting their hands in the warm, cool water?
Yeah.
And what did you experience?
So when I was putting my hand in my left hand into the warm water, it felt warm. And then the right hand, it felt cold. But then like after a while, when I started to like wiggle my fingers, I didn't feel how warm the water was. So like after 2 minutes, it said, but it said to do that with the left hand first.
But so, yeah, so the hand, so the first part, the hand and just the warm water, you feel it warm after a while. You don't feel it warm anymore. It just feels like whatever. But then you put the fresh hand in, since that's going just from the room into there, this one has not been adapting. Right? So this idea of adaptation, if you're at a certain temperature and it's not a bad temperature, it's not a harmful temperature, eventually you're not going to even respond to it. You're going to respond when there's a change in temperature. What happened when you had one hand in cool, one hand in warm, and then put them both in lukewarm?
My right hand ended up feeling like colder, and then my left hand felt warmer.
So, I mean, I find this like just surreal. Like when I do it every once in a while just because it's such a kind of cool, weird feeling of both hands are in the exact same water wiggling around and they're having such a different experience. You know, it's a pretty weird, surreal thing. I mean, well, yeah, hopefully, yeah, it'll be interesting to see how many people end up getting through all this stuff by the time we actually get to the lab exam. At this point, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, we have exactly half of them turned in. So, and actually, okay, so now, which other one? The other ones we did were the vision. You know, you should definitely know what it means to be nearsighted and farsighted or have astigmatism or how we test for things like that. How would you test somebody's visual acuity?
With that chart.
That chart, you know, it's got a special name. It was like the Snellen eye chart. And what would it mean? Let's say we found out somebody is 20/40 with their vision. What would that actually mean?
So what somebody can see at 20 feet, that person can see at 40 feet. I feel like I'm mixing that up.
You're mixing it up.
What that person can see at 20 feet, someone with like normal vision would be able to see at 40 feet.
Right. So, because remember, normally when you're actually doing this, and again, we didn't have the physical experience of it, but normally you are literally standing 20 feet from the chart and you're looking at it and trying to find out what are the smallest letters you can read without making a mistake. And then that is what you're seeing at 20 feet. And this is somebody with normal vision. How far from the chart would they be standing right now to see with the same visual acuity as you? So 20/40 means you're standing 20 feet, but a normal vision person could be standing 40 feet away and they'd see just as sharply as you. So, you know, your eyes are not as good. Unless you put on corrective lenses, and corrective lenses typically I see Liz has glass. Did you have a chance to do this one yet, Liz?
Yeah, I did it. These are for reading though. So like I didn't, if we did it 6 feet away, it doesn't, and they're not very strong.
Oh,
So I did it without them because
Right.
with them, I can see worse distance.
Yeah, no, reading glasses are not corrective lenses. Reading glasses are more like magnifiers.
Yeah, that's what they are.
Okay, yeah, no, I thought maybe they were. Okay.
So when you have like a vision test, I haven't had one in a long time and I should, but do they say in your right eye you're 20/40 and your left eye you're 20 something?
Yeah, no, each eye has its own
Each eye is different.
prescription, both
Yeah.
in terms of acuity as well as in terms of astigmatism. So yeah, if you look at your prescription, it has kind of the curvature that you need for each eye to correct it to 20/20, as well as the rotational stuff for the astigmatism.
So to test for the astigmatism, that would be more like when we brought an object close.
No,
That's not the eye chart, right?
it was another eye chart. That one that looked like an asterisk.
Oh, with the lines.
Yeah, the lines that with all the radiating
That
out.
one was hard. I found that hard for me because it was like I'd be concentrating in the middle, but then like, I don't know.
So typically if you use one eye versus the other and just kind of look at it, usually some axes look thicker or clearer or something than other axes. And it doesn't, and it's not the chart. You can take the chart and turn it sideways and the axes are still exactly what you saw before. It's not about the paper, it's about your eye. So, but yeah, so you use that. I can, we share screen again. Let me.
Professor, can I ask a quick question before I leave?
Sure.
I just wanted to know about the lab exam. Is that going to be on Thursday as well, or is that going to get pushed back?
Ugh. Let me think about it.
Well, we're doing the other one in person, so then we'd
Yeah.
have to like all come home
Yeah,
and do
no, yeah, so I would say the lab exam. In fact, the lab exam is not going to be on Tuesday. I mean, not going to be on Thursday. It's because we're going to do the rods and cones lab on Thursday. Good point. I'm looking at this thing. I don't have. This is all going to work out. I think I'll figure it all out. Maybe we'll have the lab exam in the little divot between cardiovascular, EKG, and blood pressure. That might be the best place to put it because we don't have anything scheduled for the lab and it's in the future.
Is there any way you could just like put it up and then we could take it whenever we like have a second within obviously a time range?
No, because then I find, it opens up too much of a can of worms for people sharing stuff and things.
Okay, sure.
So, but yeah, I'd say most likely, Paolo, it's probably going to be the week after it's scheduled.
Okay, thank you.
Okay. All right, and so let me go back to question about there it is. Blah, 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 vision. Yeah, so this thing. And again, and it's kind of subtle. But I can definitely say, I can say like 2 and 8 for me. They're kind of blurrier and lighter than 12 and 6. And that's just the truth for my eyes. And some people don't have much astigmatism at all. And corrective lenses are typically round to accommodate that as well. In fact, and even contact lenses they're weighted, so they rotate down. So there's a bottom and a top to a contact lens even. So there was vision. Then the other ones we just talked about. So like, for the hearing stuff, you definitely need to know about conductive versus sensory neural loss, and those tests and what they mean. Hopefully by then you'll have gone through those little videos and kind of sat down and wrote down, what were the results for these videos. And again, they're short, but they're good because they slow you down and you have to okay, I see how the test works and I see how to interpret the test. And then you're writing that in your lab notebook too. So you've got that information waiting for you when you're trying to remember what's going on there. So, are there any other questions about this stuff?